Namaste all of you. Welcome back to An Article a Day Creates an Excellent Way, the smart way to enhance your vocabulary and expressive skills. My name is Shushmita Panda, student of class 12, Kendriya Vidyalaya Malkangiri, going to read out today's article. So watch and enjoy. Today's article is about biotechnology and its applications. Introduction. Biotechnology is technology that utilizes biological systems, living organisms, or parts of this to develop or create different products. Brewing and baking bread are examples of processes that fall within the concept of biotechnology, use of yeast, living organism to produce the desired product. Such traditional processes usually utilize the living organisms in their natural form or further developed by breeding, while the more modern form of biotechnology will generally involve a more advanced modification of the biological system or organism. With the development of genetic engineering in the 1970s, research in biotechnology and other related areas such as medicine, biology, etc. developed rapidly because of the new possibility to make changes in the organism's genetic material. Biotechnology deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants and animals. The applications of biotechnology includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment and energy production. Agriculture. Alternative of conventional farming. Use of genetically modified crops is a possible solution. It is an alternative path to conventional farming. The understanding of genetics could enable farmers to obtain the maximum yield from their fields and to minimize the use of fertilizers and chemicals so that their harmful effects on their environment. Genetically modified organisms, that is denoted by GMO. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are genetically modified organisms. GMO. GM plants have been useful in many ways. Genetic modification has made crops more tolerant to abiotic stresses, that is cold, drought, salt and heat, reduced reliance on chemical pesticides, pest-resistant crops, help to reduce post-harvest losses, increased efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil. And at last, enhanced nutritional value of food. Example, golden rice, that is vitamin A enriched rice. Genetically modified biopesticide. Bt toxin is produced by a bacterium called Bacillus thuringiensis, that is Bt for short. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need of insecticides, in effect created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato and soybean etc. Bt cotton. Some strains of Bacillus thuringiensis produced proteins that kill certain insects such as lepidopterans, tobacco budworm, armyworm, coleopterans, that is beetles, and diphterans, that is flies and mosquitoes. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contains a toxic insecticidal protein. Now, pest-resistant plants. Several nematodes parasitize a wide variety of plants and animals, including human beings. A nematode, Meloidigyne incognita, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA. This process is known as RNA silencing, mRNA silencing. Medicine. The recombinant DNA technological processes have made immense impact in the area of healthcare. It enables mass production of safe and more effective therapeutic drugs. The recombinant therapeutics do not induce unwanted immunological responses as is common in case of similar products isolated from non-human sources. Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time intervals. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone. Like a proenzyme, the prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contains an extra stretch called the C-peptide. C-peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA techniques was getting insulin assembled into a mature form. Gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function and compensate for the non-functional gene. Curing adenosine diaminase deficiency, that is represented by ADA deficiency. Adenosine diaminase enzyme is crucial for the immune system to function. This disorder is caused due to the deletion of the gene for adenosine diaminase. Through gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, the patient requires periodic infusion of such genetically engineered lymphocytes. If the gene isolate from bone marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. Molecular diagnosis. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, serum and urine analysis, etc., early detection is not possible. Some of the techniques that serve the purpose of early diagnosis are recombinant DNA technology, polymerase chain reaction, and enzyme-linked immunosorbent assay, that is ELISA test. Presence of a pathogen, bacteria, virus, etc. is normally suspected only when the pathogen has produced a diseased symptom. However, the concentration of pathogen is already very high in the body. With molecular diagnosis, very low concentration of a bacteria or virus can be detected by amplification of that nucleic acid by PCR, that is polymerase chain reaction. Diagnosis of HIV. Polymerase chain reaction (PCR) is now routinely used to detect HIV in suspected AIDS patients. It is being used to detect mutations in genes in suspected cancer patients too. PCR is a powerful technique to identify many other genetic disorders. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells. It is followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by presence of antigens, that is proteins, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen. Now, CRISPR technology. The full form of CRISPR is clustered regularly interspaced short palindromic repeats. It is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segment known by CRISPR arrays. The CRISPR arrays allow the bacteria to remember the virus. If the virus attacks again, the bacteria produce RNA segments from the CRISPR arrays to target the virus DNA. It is being explored in research on a wide variety of diseases, including single gene disorders such as cystic fibrosis, hemophilia, and sickle cell disease. Transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Although over 95% of all existing transgenic animals are mice, they have numerous utilities. Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. Example, study of complex factors involved in growth such as insulin growth factor. By introducing genes from another species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as models for human diseases so that investigation of new treatments for diseases is made possible. Today's transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA. This DNA codes for a particular product such as human protein alpha-1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk. The milk contained the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Chemical safety testing. This is known as toxicity or safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances and the effect studied. Toxicity testing in such animals will allow us to obtain results in less time. Biotechnology in India. The Department of Biotechnology manages most of the programs of biotechnology sector in India. It is under the Ministry of Science and Technology. Its objectives are to provide services in the areas of research, infrastructure, generation of human resource, popularization of biotechnology, promotion of industries, creation of centers of excellence. Implementation of biosafety guidelines for genetically modified organisms, recombinant DNA products, and biotechnology based programs for societal benefits. Also, to establish an information network for the bioinformatics mission of India in the scientific community nationally and internationally. Genetic Engineering Appraisal Committee, that is denoted by GEAC. The GEAC functions under the Ministry of Environment, Forest and Climate Change, that is MOEF and CC. It is responsible for the appraisal of activities involving large-scale use of hazardous microorganisms and recombinants in research and industrial production from the environmental angle. This committee is also responsible for the appraisal of proposals relating to the release of genetically engineered GE organism and products into the environment, including experimental field trials. GEAC is chaired by a special secretary or additional secretary of MOEF and CC and co-chaired by a representative from the Department of Biotechnology. Recently, the Society of Biotechnology in India (SBPI) promoted transformation changes and approaches towards core research in modern biotechnology so that the outcome could lead to more products and technologies for economic and social gain. Here are the programs undertaken in agriculture: Wheat Genome Sequencing Program, Rice Functional Genomics, Crop Biofortification and Quality Improvement Program, National Plant Gene Repository Program, Next Generation Challenge Program on Chickpea Genomics. Utility in public health and nutrition, addressal of micronutrient deficiencies, severe acute malnutrition, food fortification, probiotics for human health and well-being, food safety, molecular detection of GM traits in foods. Development of low-cost foods, supplements, and utilization of agricultural residues for value-added products. Capacity building in food science and nutrition biology. Conclusion: applications of biotechnology could be a major tool for development in all countries. Entwined with culture and socio-ethical values, biotechnology could be utilized in solving future problems like food and water insecurity that impede national development and threaten peace in the developing world. The special words that have been collected from article are here with their meanings. Impede, delay or prevent someone or something by obstructing them or to hinder. Entwined, closely connected or unable to be separated. Phenylketonuria, a birth defect that causes an amino acid called phenylalanine to build up in the body. Cystic fibrosis, genetic disease that causes persistent lung infections and limits the ability to breathe over time. Transgenic, a transgene is a gene that has been transferred naturally or by any of a number of genetic engineering techniques from one organism to another. Array, display or arrange things in a particular way. Snippet, a small piece or brief extract. Complementarity, a relationship or situation in which two or more different things improve or emphasize each other's qualities. Mutation, the way in which genes change and produce permanent differences. Pathophysiology, the study of changes in the way the body works that result from disease. Embryo, an animal that is developing either in the mother's womb or in an egg, or a plant that is developing in a seed. Recombinant, relating to or denoting an organism, cell or genetic material formed by recombination. Eukaryotic, any cell or organism that possesses a clearly defined nucleus. The eukaryotic cell has a nuclear membrane that surrounds the nucleus in which well-defined chromosome bodies containing the hereditary material are located. Infestation, the presence of an unusually large number of insects or animals in a place, typically so as to cause damage or disease. incognita in a way that conceals one's true identity. Nematodes, a worm of the large phylum Nematoda such as roundworm or threadworm. Biopesticide, a pesticide consisting of naturally occurring or genetically engineered microorganisms such as bacteria. Abiotic, physical rather than biological, not derived from living organisms. Biopharmaceuticals, a biological macromolecule or cellular component such as a blood product used as pharmaceutical. Therapeutics, a branch of medical science dealing with the application of remedies to diseases. Thank you for watching.